So it's 2:30, and I think I'm going to slowly get started here, assuming that a few more people will be joining us in the next couple minutes. So welcome to the symposium on looking at aging biology as a phenomenon of complex dynamic systems. There are 6 of us who are co-organizing this workshop. So we've got myself, Nan Hao, Morgan Levine, Marcel, Olde, Rikkert, Scott McIsaac, and Nick Strustrup. And each of us will be giving a talk, which will last about 15 minutes plus 4 for questions, 3 today and 3 tomorrow. So it's a workshop that's in 2 parts over 2 days. And those talks will then be followed by about 40 minutes of discussion on each day. So the background for it is that we're all people who in one way or another are working in biology and have in the field of aging and have in one way or another realized that a lot of the biological approaches don't sufficiently use complex systems theory, complex systems dynamics, complex systems approaches to tackle the problem of aging. And I think this is probably general to biology, but more specifically to our field of aging biology. And I think we probably also feel, though, that a sea change is coming. In the last couple of years, especially, things are starting to turn around. And so it's kind of the moment to try to join forces, figure out what we can do to push people working in aging biology to really start thinking in a complex systems framework, adopting those methods. And what are the ways we would ask questions? What are the questions we should be asking? So the goal over the next couple of days is to figure out how to do that. So it's not just a series of talks. The idea is really to produce a research agenda and a position paper and to figure out if we want to organize ourselves further, have other meetings. So that's really the goal over these 2 days. I think it's crucial that we're doing it here at ICCS because a lot of us probably don't come from formal complex systems backgrounds, but we've come more to the field for other reasons. And I think it's important for us to be drawing on the expertise of researchers whose main expertise might not be aging or might not be biology, and see what you have to contribute to how we can think about the problem of aging from a complex systems perspective. So really, that's the goal over these couple days. The structure is that we have, as I mentioned, 2 days with 3 talks per day followed by discussion. The idea here is that these talks will give diverse examples of how complex systems approaches can be used to understand aging. So the 6 of us have actually quite different backgrounds from within biology, from working on yeast, really at a cellular molecular level, up to demography or clinical applications. So we'll see some very diverse examples, but it's probably just a very tiny bit of what could be done or could be imagined. The first day's talks will look more focus on aging as a complex systems problem. And the second one will more look at approaches from complex systems theory to look at aging. And the discussion on the first day will be more geared towards a research agenda, and on the second day towards a position paper. So obviously there's a lot of overlap between those. It's not a hard and fast distinction, but we've tried to set it up that way. So without further ado, I'm going to let our first speaker get started. It's Nan Hao from the University of California, San Diego. So Nan, you can share your screen, and looking forward to hearing your presentation. You're still on mute there.
Hold on a second. Okay, so first I want to thank Alan for organizing this workshop. He really did all the heavy lifting to make it possible. So truly appreciate that. So today I'm going to talk about our recent work on single cell aging. And as we all know, aging is a super complicated problem. And before we ask the question why humans age, we decided to ask a relatively simpler question, but related question, why cells age? So many damage and stress factors have been identified to contribute to cellular aging, such as chromatin instability, loss of proteostasis, reactive oxygen species, mitochondrial defects, and many others. It is generally believed that all these damage factors accumulate gradually during aging and leading to cellular declines and eventually cell death. However, in each individual cell, how these damage factors act together to drive the single cell aging processes remains unclear. For example, one possible scenario is that cellular aging is driven by many independent damage factors that accumulate stochastically with different rates, resulting in different types of cell deaths in different cells. In another scenario, cellular aging could be driven by an integration of all the damage factors, leading to a common aging and death pathway. A careful single cell analysis is required to determine which scenario actually underlies single cell aging. To this end, we studied the replicative aging of budding yeast Saccharomyces cerevisiae, which is a genetically tractable model for the aging of mitotic cell types in mammals, such as stem cells. And the replicative aging in yeast is measured as the number of mother cell divisions before death. So to track the molecular and the cellular changes during aging, we developed a microfluidic device. This device traps single mother cells at the bottom of finger-shaped chambers, allowing them to bud continuously, while the daughter cells are pushed out automatically. There is also a small opening at the bottom of the chamber, allowing daughter removal even when the mother cells switch budding directions. So let's further zoom in at a single chamber and look at the whole aging process. This is the mother cell. It starts budding upward and then switch budding direction, budding downward, and switch again, start generating these super elongated daughters, meaning they are truly old, and eventually they will stop division and eventually die. Okay, so using this approach, we found that isogenic cells showed two very distinct phenotypic changes during aging. And about half of the cells, when getting old, continuously produced elongated daughters until death, while the other half of the cells produce small round daughters until death. We called them mode 1 aging and mode 2 aging. A slowdown of cell cycle rate is a major phenotype for yeast aging. And we found that mode 2 aging features a more dramatic slowdown of cell cycle rate and a shorter lifespan than mode 1 aging. And aberrant structural changes in nucleolus and mitochondria are major hallmarks of aging in yeast and in many other organisms. To determine the mechanistic basis of mode 1 and mode 2 aging, we tracked the morphologies of these organelles. And we found that the nucleoli were dramatically enlarged in all mode 1 aged cells, but not in mode 2 aged cells. While mitochondria in mode 1 cells remains a normal tubular structure throughout the entire lifespan, whereas those in mode 2 cells became aggregated before death. So these results really indicate that different modes of aging specifically target different organelles. Mode 1 aging leads to nucleolar decline, while mode 2 aging leads to mitochondrial decline. Nucleolus is where the rDNA, ribosomal DNA, is transcribed, and the ribosomes are assembled. Previous studies showed that age-associated nucleolar enlargement is related to the instability of rDNA located in the nucleolus. And a major mechanism to maintain the stability of rDNA is chromatin silencing, mediated by a conserved lysine deacetylase, SIR2, which is encoded by the best studied longevity gene today. To monitor the rDNA silencing in single cells, we inserted a fluorescent reporter gene under a constitutive promoter at the non-transcribed spacer region of rDNA. Because the expression of this reporter gene is repressed by silencing, so a decreased fluorescence indicates enhanced SIR2 activity and silencing, while an increased fluorescence indicates reduced SIR2 activity and loss of silencing. Then for mode 2 aging, we consider the proposal in human aging that the deficiency of heme, a major functional form of iron, may play a key role in mitochondrial decline. In yeast, heme activates the HAP transcriptional complex to maintain mitochondrial biogenesis and function. To monitor the HAP activity during aging, we used an infrared fluorescent reporter gene, the fluorescence of which depends on a heme degradation product, biliverdin. Therefore, a high IRFP signal indicates a high heme level, while a low IRFP signal indicates a low heme level. So to determine how the SIR2 and HAP pathways operate together to drive the aging process, we introduced both the rDNA GFP reporter and the nuclear anchored IRFP reporter in the same cell to simultaneously track rDNA silencing SIR2 activity and HAP activity in the same cells to determine how they drive aging process. Here in this slide, I want to show you representative movies of single aging cells and the trajectories quantified from this movie. Here in this 3D space, the z-axis represents percentage of lifetime from 0 to 100. The x-axis represents the nuclear IRFP signal, while the y-axis represents rDNA GFP signal. Let's start with mode 1 aging cells. And as we can see, during aging, cells undergo gradual increase of rDNA GFP signal and an increase of nuclear IRFP signal. So now let's look at a mode 2 aging cell. Very different from mode 1 cells, we see a sharp depletion of IRFP signal and a gradual decline of GFP signal. Now let's look at the whole population of isogenic cells. And here, the red points are from mode 1 aging cells, and the blue points are from mode 2 aging cells. We can further project the initial states of all the cells onto a 2D plot. Here, to remind you again that because SIR2 repressed the expression of rDNA GFP by silencing, a high GFP signal indicates a low SIR2 activity, while a low GFP signal indicates a high SIR2 activity. And in the 3D space showed the whole aging process of all the single cells, and we can further project the ending states of all the cells onto another 2D plot. As we can see here, isogenic cells began their life with relatively uniform levels of 2 reporters. During aging, they diverged quite earlier in life and progressed towards 2 opposite ending directions with anti-correlated SIR2 and HAP activity. Mode 1 aging cells ended their life at a state with low SIR2 and high HAP activity, while mode 2 cells ended their life at a high SIR2 and a low HAP state. So these results really indicate that genetically identical cells can age towards 2 discrete ending states with anti-correlated SIR2 and HAP activity, suggesting a potential mutual inhibition between SIR2 and HAP that may mediate the earlier divergence of aging processes. We further confirmed this mutual inhibition by genetic analysis. And in addition, both SIR2 and HAP also have positive autoregulations from previous literature. So based on this circuit, we developed a simple computational model composed of 2 ordinary differential equations incorporating mutual inhibition as well as positive autoregulation of SIR2 and HAP. To understand how this system works, let's start with the system without mutual inhibition. Now, SIR2 and HAP form two independent positive feedback loops, generating bistability of each variable, resulting in 2 times 2 equals 4 steady state. So on this SIR2 and HAP phase plane, the blue horizontal lines are SIR2 narclines. The red vertical lines are HAP narclines. Where they intersect, the open circles are unstable fixed points, and the closed circles are stable fixed points. We have now 4 stable fixed points. And when the mutual inhibition is added, the stray narclines are distorted. The mutual inhibition prevents the narclines from intersecting at this high SIR2, high HAP state region, eliminating one of the stable fixed points. To address the stochastic nature of single-cell aging, we then switch to a stochastic version of our model and computed a potential landscape by numerically solving the corresponding 2-dimensional focal plane equation. And each of the stable fixed points of the deterministic system corresponds to a local veil on the landscape, with the depth of the veil reflecting its fate decision probability. The deeper the veil, the higher the probability. And in this way, single-cell aging processes can be viewed as a ball rolling down toward one of the veils on the landscape, with the low SIR2 high HAP veil corresponding to the ending state of mode 1 aging, while the low HAP veils corresponding to the ending state of mode 2 aging. We then examined the genetic perturbation of the circuit, and among which twofold overexpression of SIR2 provide very interesting results. In addition to mode 1 and mode 2 aging, SIR2 overexpression generated a new mode of aging that ended at high SIR2, high HAP state. We call it mode 3 aging. And these mode 3 cells turn out to be very long-lived, with a lifespan about twice of the average lifespan of Y-type cells. And in addition, these mode 3 cells can retain a normal cell cycle rate throughout their entire lifespan, very different from mode 1 and mode 2 cells. In our model, if the SIR2 level is increased by twofold, the increased SIR2 activity can partially counteract the inhibition from HAP, leading to the emergence of a fourth stable fixed point on the phase plane and a new veil on the landscape. And the progression towards this high SIR2, high HAP stable point corresponds to the long-lived mode 3 aging observed experimentally. However, at the same time, SIR2 overexpression biased the fate decision towards this high SIR2, low HAP state, which is consistent with our model observation, with our experimental observation that a large fraction of cells underwent short-lived mode 2 aging. As a result, the average lifespan of the whole population was only extended very modestly under this condition. We speculate that by reducing the proportion of cells that undergo mode 2 aging will further extend the lifespan of the whole population. And based on our model, an elevation in the HAP level can move the high SIR2 low HAP stable point and an unstable point closer to each other, and thereby reshape the aging landscape to bias towards high HAP states directing more cells to mode 1 and mode 3 aging, and thereby a longer lifespan. To test this model prediction, experimentally, we overexpressed both SIR2 and HAP. And consistent with the model prediction, very few cells underwent mode 2 aging, while a large fraction of cells experienced long-lived mode 3 aging. And as a result, the combined perturbation can effectively extend the lifespan of the whole population, much more dramatic than overexpression of either SIR2 or HAP alone. And in addition, the combined perturbation can enable cells to retain a relatively normal cell cycle rate, promoting a healthy longevity of cell. So to sum up, let's step back and look at our original question. How do single cells age? It turns out that neither of our initial models is correct. Cell aging is not driven by random accumulation of multiple independent damage factors or an integration of all the damage factors. Instead, our results showed that cellular aging can be considered as a fate decision process. Quite earlier in life, a cell has to make a decision on whether they want to suffer silencing loss and nuclear decline while keep their mitochondria healthy, or they want to suffer heme depletion and mitochondrial decline while keep their nucleoles healthy. And once they make this decision, they will stick to it throughout their entire lifespan until death. And in this way, based on the circuit dynamics, single cell aging processes can be viewed as a divergent progression towards different wells on a SIR2 and HAP landscape. And importantly, guided by model-guided genetic perturbation of the circuit, we can reshape the aging landscape and enrich a new mode of aging process that is super long-lived and not observed in Y-type cells before. So this really raises the possibility of rationally designing intervention strategies that target multiple network nodes instead of single genes to reprogram how cells age and to more effectively promote longevity. So finally, for acknowledgement, the work is primarily done by a very talented postdoc in lab, Yang Li, and especially also for the theme of this conference, I want to thank my collaborators. We have assembled actually a very multidisciplinary team at UCSD to study aging. And in the team, we have engineers, we have physicists, we have computer scientists, we have, of course, biologists, that we all share the common interest in the fundamental biology of aging, and we work very closely together to study the aging process. And especially our collaborator, Professor Jeff Hasty, who is a physicist, Professor Lauren Pilas, who is a molecular biologist, and Professor Lev Severin, who is a modeler, and many other trainees. And finally, our funding sources, and thank you very much for your attention.
Thank you, Nan. I'll give you some applause, even if we can't hear it from everybody.
Thank you.
Let's take time for just one quick question. Try to keep us roughly on schedule.
Okay.
Does anybody have a question? Okay. Let's see. I'm going to let Scott.
Alan,
Scott
go ahead.
Yeah, Scott has a question here, so I'm going to let him jump in there.
Oh, there we go.
Okay.
Hey, Nan, great talk. Hey, how you doing?
Good.
I had a question about this. So you showed this nice mutual inhibition between HAP and SIR2. And I was curious if you guys are thinking that's SIR2 is really the only thing that's important there, because one of the surprising things we found was that deletion of UBR2, which upregulates the proteasome, also inhibits the nucleolar instability.
Mhm.
It looks a lot like a FOB1 delete, which directly impinges on the rDNA locus.
Mhm.
So I was just curious if you're thinking or exploring if there's other interactions going on, or if SIR2 really is just the only one. Are there multiple routes in a sense?
Definitely. I think although we focus on SIR2 in our study here, but SIR2 is, I will be very surprised that SIR2 is the only major regulator. And as you said, UBR2 is one of them, and I imagine there are many other factors that contribute. I think it is more of a pathway or a network that regulate nucleolar structure and function that may contribute to that. And SIR2 is one of the regulator there, and there are many other regulators we will imagine, yes.
Okay, thank you. I think I'm going to take just a second, use my prerogative as the organizer to give a commentary on Nan's talk as well, which I think is just fantastic. I think this illustrates really well why the complex systems perspective is crucial here. So if we're looking at this as single genes that are having an impact, we're going to completely miss out on both the emergent states. So these different modes, I think from a complex systems perspective, we can look at as emergent states of the cells. And also on the need to be considering the interactions through a network perspective. So I think that's just really exciting work. And I'm going to now give the microphone over to Morgan. Nan, could you stop share?
Yeah.
Let me hide this box. All right, thanks again to Alan for really kind of taking the initiative to organize this whole session. I think it's really exciting, and I think it's really good now. A lot of people have been talking about, how to use complex systems theory and methods to actually study aging, but I think it's really great that there's now this initiative of groups of people to do that. So to kind of jump in, hold on, I can't see the tops of my slides because of this. Okay, well, whatever, I'll just try and remember what was there. I kind of want to start with the way I think about aging, or at least the concept of aging. So I like to think of it in terms of these kind of multiple levels of biological organization. So you have aging that really is starting at these lower levels of organization, and then it's really working its way up. And this is, as Alan kind of pointed, an emergence of the changes that we're seeing. And these changes can happen for a variety of different reasons. Some of them can be just random errors or kind of entropically derived changes. I think another potential source of these changes are adaptations or compensations that arise with aging that I think are often overlooked. We like to think of kind of this damage accumulation. We don't actually think of this also as kind of an adaptive process as well. And then there's also obviously selection that is happening at not just the organismal level, but also the cellular level that could also drive kind of changes in the overall system. So basically, Alan alluded to emergence before, but basically life is an emergent property of all the interactions happening at these lower kind of biological levels of organization. And really what we think of as the manifestations of aging, so disease, functional decline, are a direct result of the changes that are happening at these lower levels. And for that reason, it's really important to kind of study aging from kind of a bottom-up perspective. Can we figure out what's happening to the system at the lower levels and how that gives rise to some of what we see at the higher levels? So there's been decades of work really going into kind of the molecular and cellular changes that we observe with aging. So these come down to things like epigenetic modifications, genomic instability, mitochondrial dysfunction, stem cell exhaustion, telomere attrition, loss of proteostasis, et cetera, inflammation, senescence, changes to the extracellular matrix. And together, these actually will then give rise to changes in tissue and organ integrity, and ultimately give rise and influence pathology of disease. So an area that I work mostly in is in epigenetics. I like to think of the epigenome as the molecular operating system. So basically the epigenome is really what's underlining cellular phenotype. It's giving instructions to the cells on how they should behave, how they should respond. And it's involved in a number of different processes, from cell proliferation, differentiation, transcriptional repression or activation, genomic imprinting, organization of chromatin, and X inactivation. And for those of you not in biology, basically your epigenetics is gonna dictate what type of cell, what the features of a cell. So obviously in general, all your cells have the same DNA, but the epigenetic modification are basically making a neuron and a fibroblast a fibroblast. So what I mainly focus on in my lab is something called methylome. So this is one type of epigenetic modification. It's involved with chemical modifications to these CpG sites, so these cytosines that are followed in 5' to 3' by guanine. And these have a propensity to become methylated with, and basically you have 28 million of these CpGs throughout the human genome. On average, about 70% of them tend to be methylated, except something that was observed maybe a few decades ago and is really fascinating is that the pattern, or what I might even consider the program, is actually changes quite dramatically with age. And for that reason, people have actually, so here's an example kind of this. This is just a distribution of the level of methylation in a group of cells. This is from blood for kind of prenatal samples, middle-age samples, and samples from individuals who are 70 and older. And as you can see that you get a shift in this distribution with age. And on the right-hand side, this is basically showing for, I think, 450,000 of these CpG sites genome-wide, the change with age. So this is a slope in changes in methylation with age plotted against the p-value. So as you can see, there are dramatic changes in DNA methylation with age. And a really interesting thing is that these don't seem to be random. There do seem to be some CpGs that are much more prone to these changes with age. So much so that people have actually tried to use these changes to quantify aging or measure what we call biological aging. So can we actually measure this system and use it to actually estimate the agedness of a sample? So whether it's a tissue sample, cell culture sample. And the idea is that, you know, having these what we call biomarkers of aging or measures of biological age is that they can actually serve a purpose in clinical trials as endpoints to actually, when we're trying to modulate the pace of aging. They might inform some basic biology of what drives aging. And for epidemiologists or people in public health, they also can be used hypothetically for risk stratification. So the goals of kind of these quantifications are that can we actually measure the system and not just differentiate between the system of a 20-year-old and an 80-year-old, but also can we actually understand the difference between two 20-year-olds who might have different kind of future health risks. So these biomarkers that have been developed are called, have been traditionally called epigenetic clocks. So again, because of the precise age changes, people have used machine learning to predict the age of a sample. So you can take, for instance, a blood sample. We can measure methylation right now at billions of different sites. And we basically use a supervised machine learning approach to develop an age predictor based on those methylation patterns. So a number of these epigenetic clocks have been developed starting since 2011. And the interesting thing that we've found is that your difference between your predicted age and your chronological age tends to map on to a number of different aging outcomes that we think are important, but it's not the same for every clock. So there seems to be some clocks that are better at predicting certain conditions and some clocks that might be better at predicting other conditions. And really, kind of what we think is going on is that different clocks actually seem to be capturing different pieces of this overall program, or different kind of biological processes or phenomena. So this is from a paper that's currently under review by one of my former postdocs, Ian Liu. So basically, this is just showing, as a function of chronological age, 3 different clocks measured in a bunch of different tissues. So each color here represents a different tissue. And as you can see, the clocks actually pick up fairly different, they pick up differences between tissues in different ways. So the original Horvath clock was trained to actually not show tissue differences, whereas these 2 clocks were trained in whole blood, but actually do show, do track with age in other tissues, but you can see their intercepts and slopes are somewhat different depending on what tissue you're looking at. They also are different in terms of picking up things like tumor versus normal samples. So again, these are 6 different clocks in this case, and as you can see, some of them are better at differentiating normal tissue from tumor, whereas others actually go in the inverse direction and suggest that there's a deceleration in tumor versus normal. And finally, we can actually look in tissue culture, and we can senesce cells and compare early passage to either oncogene-induced senescence or replicative senescence. And you can again see that certain clocks are picking up these changes differently than other clocks. And really what this comes down to is that we think that these different clocks are picking up different parts of this program. So again, epigenetic aging, or these changes that we're observing in methylation levels with CpGs, could be due to a variety of different factors. Again, some of them might actually just be entropic, so what we call epigenetic drift. Some of them might be compensatory changes to the cells. Some of them might be cellular selection. And ultimately, these also might have different downstream implications for function. So whether they're impacting cell proliferation, whether they're impacting cell identity, or also kind of transcriptional repression and activation. So what my lab is really interested in trying to tackle this problem is not to actually look at the clocks as a whole, because what we think is that they're picking up actually a bunch of different pieces of different systems. But can we actually kind of decompose the signal and figure out what the individual components that are captured in the different clocks actually are? So essentially, can we identify the parts of the clocks and then see how they map on to different aging outcomes or change differentially within different tissues and cell types? So this is kind of a theoretical example where you have an actual clock, and can you find the different pieces that make the actual clock run? So this work is really preliminary, but I'm going to show just a tiny bit. So this is one of the current postdocs in my lab who's working on this, Dr. Albert Higgins-Chen. So basically what we're doing is we're applying different network analyses techniques to identify what we call co-methylation modules. So these are basically CpGs that seem to change together across a bunch of different either cellular experiments or in a bunch of different tissues. And if they change together and they have a lot of kind of shared connected nodes, we think that they're part of one singular system. So for now, what we've done is we've started looking at just the CpGs that have been implicated in any of the existing epigenetic clocks. And from there, we can actually branch out and explore other CpGs that are not part of the clocks. So far in this work, we've identified 15 of these co-methylation modules. And what we can do is actually look at their influence within the different clocks. So which ones are actually driving the different clock scores? And is that what's relating to the differences in the clocks in terms of what they can pick up and what they can predict? So we're color coding these modules. So this basically shows the distribution of the influence in different modules in all of these different clocks. And as you can see, the clocks do have pretty different kind of proportions of these modules. So this Yang clock has a lot of the black and red, whereas perhaps some of these clocks have more of this green-yellow module, and these mitotic clocks have a lot more of these turquoise and pink modules that are picking up in them. So the other thing we can do is we can actually look again at the individual module pieces across different scenarios. So this is all for one clock. If you broke down just the individual module pieces, and in blood, we can compare samples from young individuals to samples from old individuals. And what you can see is that actually not every module is picking up the age difference in blood for young versus old. You have some modules like this one, which is what we call the brown module, and then this gray module, which is really differentiating young and old, whereas these other modules like the red one and purple, you actually don't see much difference, at least in blood, for age. We can similarly compare different tissues, and again, you see different mapping of kind of these differences depending on which module you're looking at. And also going back to kind of senescence or actually cell culture experiments, where again, some modules are picking up these signals where others aren't. So we actually think that if we can, repeat this over a bunch of different experiments, then we can actually start to decompose and understand what the different pieces of these clocks are picking up. So, in moving forward, really a goal is hopefully to actually be able to do more single cell analysis. So right now that's kind of been a struggle with DNA methylation. But really what we want to understand is on a single cell, within a single cell, which CPGs are actually co-methylated together and show patterns across individual cells. We also think that we need to move more towards this bottom-up approach. So rather than determining just which CPGs are changing with age, can we actually connect these changes to specific cellular, molecular, and physical features, and from there actually build out these networks. And ultimately, part of this will be actually investigating cause and effect. So what DNA methylation processes are modifiable and by what? And what is the impact of altering DNA methylation for specific sets of CPGs? So with that, just in conclusion, basically what we see with aging is the emergence of changes at lower levels of biological organization. And DNA methylation, which is one form of epigenetics, is a molecular feature. This strongly changes with age and has direct implications for cellular phenotypes. Again, we like to think of this as kind of the operating system of the cell. Epigenetic clocks were developed to quantify something called biological age, yet at the same time, they actually capture very diverse kind of systems depending on which clock you're looking at. So either with different causes for these changes in methylation or different effects of the methylation changes. And really, in order to understand the system, we need to better determine what processes these different types of methylation changes are reflecting. So with that, I want to thank the people in my lab. This is an outdated picture, so we're missing a lot of people, and with COVID, we can't take a new one. And also my collaborators and my funding at NIA. And again, Alan, thank you for organizing this session.
Thank you. And again, I'll do the one-person clap. So yeah, thanks. I think we have time for a couple questions here. I forgot to mention last time, you can't just start talking. So let me know in the chat if you have a question, and I'll unmute you. Okay, so Lou Lipsitz has a question. Just a second here. Go ahead.
Hi, Morgan. Can you hear me?
Yes.
Yeah.
Very interesting work here. I always enjoy listening to you. But I would like to just take minor issue with a couple of things. First of all, the comment that the life is an emergent property due to interaction of processes at lower levels. Being a clinical investigator myself in the field of complexity science, I would argue that, societal levels, environmental levels, very much larger levels also influence our lives. The other comment is just the effort to decompose the signal to identify the parts is very sort of reductionistic, very linear, and not really as consistent with complex systems as we might like. So I think it's very creative and very interesting to see these modules, but I think what you're going to find is even more informative is the interaction between those modules, because that's what creates the emergent properties. It's not any one of them, but how they interact together at all different levels, not just the lowest levels, but how those cells form organs and eventually people and societies are really what's important. So just a couple of comments to think about.
So I totally agree with both your comments. So yes, we need to also, I did stop at the organismal level, but there are obviously levels above that, and that there is a feedback loop. But the way I think about it is even that way that gets integrated is still at the lower levels and then feeds up, even if it is, you know, something coming from a higher level. And then in terms of the kind of making this reductionistic, I think the issue with epigenetic clocks is people have said, you know, we don't understand what they're representing. And I think in terms, first we have to actually figure out the pieces, right? The nodes of the network. And right now I think we don't even know what these nodes are. And in my lab, what we like to talk about them is they're fuzzy modules. They're not independent modules that are completely disassociated from each other. They connect with each other. And I totally agree with you that it's going to be really important then to take this and piece it back together. So actually some stuff we've done that I haven't talked about is, can you then look at individuals and cluster them based on how they're changing in different modules? And then that kind of gives you the idea of emergence. So people who tend to develop some clinical manifestation tend to have accelerated aging in maybe 4 of these modules, right, but not other ones. So totally agree with you. We don't want these pieces, but I think we need to first be able to define the pieces before we can start really building and putting it back together. But yeah, great comment.
Challenge. Good. Thanks.
Thanks, Morgan. I have one more quick question here from Robert, which is just, is there any source code available for the various clocks? I imagine yes.
There will be once, yeah, this is very preliminary, but yeah, we're happy to, we'll put everything out. Yeah.
Thanks. So yeah, once again, I kind of knew you were thinking along those lines and appreciate Lou's questions, because I think that succeeds in tying it successfully into everything else that we're doing here. Shall we move on to Marcel's talk? Thank you very much.
Okay. Thank you, Alan. Hope you can see this slide now. So thank you, Alan, for having this great opportunity. And I would like to follow up on this interesting basic biological aging talks with a more clinical talk, an application in one of the biggest problems for healthcare at the moment, Alzheimer's disease. And I would like to show you, to take you with me on the complex system research challenge that's posing Alzheimer's to us at the moment. Doing this, I'm Marcel Olde Rikkert. I'm a geriatrician, a clinical researcher at the Radboud University Medical Center, and I'm also heading the Alzheimer's Center in our university. So that's my background. And I don't have any conflicts of interests here, not involved in pharma research in Alzheimer's. And I've got three short pieces of information I would like to share with you. First, looking at the paradigms that I think are changing towards complex systems ideas, and then how complex systems research methods might help us in Alzheimer's. And last but not least, where this can help us to really find, for the first time, some sort of modifying treatment in Alzheimer's. So starting with the paradigms, here you can see at the left-hand side some of these beautiful drawings of Alois Alzheimer, who in fact described Alzheimer's in 1907 with figuring plaques and tangles, the plaques between the neurons and the tangles, the tau aggregations in the neurons. And in fact, it was the primary figuring of the amyloid cascade hypothesis, though it wasn't known at that moment. And in these 100 years, of course, a lot of information was found, and you can see it a bit at the right-hand side. But still, in fact, this amyloid cascade hypothesis is leading. Most of the drug interventions are now still along this road. And here you can see a picture which seems a bit complex, but still is rather a linear process from the amyloid precursor protein down to A-beta 42 amino acid aggregation, forming amyloid plaques, and then giving aggregate stress, inflammation, and leading to neuronal death and finally to cognitive impairment. Together with the tau phosphorylation, but still you can see that's a rather linear lead pathway to this cognitive decline. So that's what in fact is still leading a lot of the pharma research. Though on the other hand, we know that most of these trials also along this road did not give any functional improvement for the patient. We now have some of these interventions that are helping to reduce the amyloid burden, such as the aducanumab now, but still they don't have a really clinically relevant improvement of the patients. So it seems that this amyloid cascade hypothesis from this perspective is not enough to clarify the picture. And there's more reasons to believe that it's more complex. So the last 30, 40 years, in 8 very good studies in high-income countries, we see an impressive reduce, a 30% average reduce in age-corrected incidence of dementia. And at these decades, in fact, there was no way of having a better amyloid modifying therapy. So that's again giving some evidence that beta amyloid cannot be more than just part of the puzzle for, in fact, modifying the pathophysiological pathways. And this is one of these 8 studies, in fact, the Framingham Heart study, which also shows data of reduced incidence and relates it to cardiovascular health, cardiovascular treatments, but also to better lifestyle, education, and a richer social context. So the high-level factors that may influence, in fact, neuronal health. But there are even more reasons to believe that it's a very complex disease. And here you can see some of our own data of a cohort of people with dementia. And you can see the upper 3 panels, which is cognitive performance on the mini-mental state, and the lower 3 are, in fact, daily functioning measures. But I just want to point out now is the large heterogeneity of, in fact, the clinical trajectories of dementia over time. And when we try to do some latent class analysis, we can more or less figure out that there are 2 classes, but still there's a large heterogeneity within these classes, and they do not solve quite the problem, which seems so heterogeneous that it's hard to believe that it's just one linear pathway explaining this. And finally, I would like to point out some of the very interesting hallmark studies SAFA did, already presented it in the New England Journal of Medicine 10 years ago, but still very worthwhile looking at. Here you can see, here you can see that in fact, when people age, you can see it downwards from 70 to 100, that on the left-hand panel, in fact, only hippocampal atrophy is dividing people with dementia and without dementia. While at the 2 right-hand panels, you see that, in fact, the neuritic plaques, the amyloid plaques and the tangles do not separate these individuals anymore. So the hallmarks of the amyloid cascade hypothesis do not make a difference between dementia and no dementia. So again, evidence that it's much more complex. And this complexity is also demonstrated when you look at other evidence, looking at other perspective on, in fact, the biology of Alzheimer's. You can see mitochondrial damage. You can also see changes in glucose metabolism, even leading to already insulin treatment trials in Alzheimer's, though last week published, in fact, in the New England was also a negative trial. And DNA methylation, as just referred to by Morgan, is also shown in Alzheimer's. And of course, already for a long time, we know that there's neuroinflammation and there's a lot of vascular damage. So all this together, in fact, points at a complex paradigm, a multi-scale, multi-causality paradigm, which is far more reasonable to accept given all this evidence. So here you come to a picture in which I would like to just schematically present this paradigm. At the left-hand side, you see a normal adult, a young adult with a cognitive reserve, free of damage in the brain. But during aging, in fact, to the right, there's an accumulation of different sorts of damage, but there's also some repair and there's some compensation. So, in fact, at the end of the row, there's so much damage in the brain that cognitive performances are so low that you can diagnose dementia. That's, in fact, what's schematically presented in this multi-causal, multi-scale paradigm. And you can see it in another way, which demonstrates the heterogeneity in these figures, the 1, 2, and 3 figure, the left hand, you can see somebody who's having risk factors that are DNA related, but also cardiovascular risk factors and lower education. Oh, it's very sensitive. And person 2 is in fact having DNA, but for instance, lack of deep wave sleep as a risk factor. So it's another combination of risk factors interacting together and giving, in this person, another shape of, in fact, dementia, another rate of decline, and also another phenotype. So that's the interacting component cause model, which is in fact following this other paradigm for Alzheimer's. So summarizing this, we have a multi-causal, multi-scale paradigm of Alzheimer, I think, which is giving additional perspectives on what we see in many trials and has to bridge one way or another the molecular, the cellular, the organ, the organism, and the social system scale. So lack of sleep, for instance, is at the organism level, but still has it impact on the molecular and cellular amyloid aggregation, which has been shown to have a real connection. Same for the social system isolation, obvious now in the COVID period, has also its damaging effect on the neuronal system. So there are a lot of bridges between these scales. So where does that lead us with complexity research methods? How do we find these bridges, and how do we in fact have profit of the complexity science methods that are already present? We're trying to work on this at the moment, and we are following this line in which we start with mind mapping. In fact, what I did just now is mind mapping of evidence that's present and that you try to shape into explanatory models, but you can do it also more in a formal way using group model building strategies. I'll come to that later. And again, a step further is trying to get this into a system dynamics computational model, which finally should have and should result in valid individual models that can be of prognostic, but also of therapeutic help. So this is a road map for complexity science in a clinical perspective. And here you can see the first results of what we did in a group model building study, in which, in fact, Jeroen Ulleman, as a PhD student, carried out a lot of work together with colleagues from Amsterdam, also colleagues from other departments and other faculties in our university. And we tried to link on the red hand, in red, in fact, the neuronal factors together with the more bodily factors, and in green, the psychosocial factors, looking at what is known from literature, but also the insight of 13 experts in different fields, connecting in discussions, in several meetings, in a formal strategy of group model building, how these connections, in fact, could be best made. So that's group model building, which brings further insight in this complexity of interacting factors. And here in such a model, you can also, in fact, deduct between the centrality and closeness centrality of the different factors by network analysis. And for instance, this closeness centrality pointing at factors that are having a lot of influence on different nodes closely by, with few mediating other factors, so they may have big impact when you address them in a therapeutic trial. So this is a way, first preliminary idea, getting an idea of what you could use as targeting variables. But you need other steps, such as the computational modeling, and that's what we are working on at the moment, trying to use longitudinal data from, for instance, the APNI study, and try to have different groups used, in fact, the decline over time, connecting with other variables to present these kind of interactions and link them to data that are already known, calibrate them, and in fact, calibrate them again and again on different subsets of these longitudinal cohorts. And then you might get a fully parameterized system dynamics computational model of Alzheimer's disease. That's what we are trying to realize at the moment. So there we use complex systems research methods in fact, trying to figure out the pathophysiology of Alzheimer's when you use different scales and multiple components as causal factors. What does it head to when you go to interventions? Well, you might consider, again, these 3 different patients with dementia with their different factors, and you can already see that you might address in the 3 different individuals also different components in this therapeutic intervention. So, for instance, in person 2, you might go to this lack of sleep and focus more on a cognitive behavioral intervention there, while at another subject, for instance, subject 1, you might will to go to the cardiovascular risk factors and put all effort on that. So you could have individualized interventions when you first figure out in the model and use, in fact, also deep phenotyping per individual. Then you could address what has most impact on each individual's dementia trajectory. And there has been already some first research using this perspective, and in fact it was a New York study of Isaacson and others, and they addressed this way of individualizing treatments and also prevention by deep phenotyping, and they compared it not directly but to a historical control group. And on the left-hand side, you see that prevention in the blue and the orange lines, in fact, did better than the green historical controls, even when there was low compliance to the intervention. And on the right-hand side, in early dementia, and individualized treatments only did better significantly when there was a high compliance. But still, it did better than the historical control. So it shows that there is already a figure of evidence for this individualizing treatments, which in fact go back to the first idea of the interacting component causes that are different for individuals. So coming to an end, Alzheimer's presents really a big challenge for our societies, for our healthcare, but also for science, of course. You can see it on the many trials that did not give a positive result so far. We might be challenged to take another perspective of a more complex, multi-scale, multi-causal paradigm, and we might evaluate and increase our insights here by using complexity science methods and computational models. But as in other perspectives and paradigms, we still need proof of concept trials that help us to show whether we can do better prognostications with these complex models and whether they really, in controlled, compared studies, deliver more positive treatment options. So that's the way to go. We'd like to do this with our Alzheimer's Center and with our geriatric departments, and also, of course, with the society that Ellen brought so nicely together. So open for some questions, and thank you for your attention.
Once again, my solo applause. And if you have a question, yeah, Andrew Rittenberg has a question, so go ahead.
Hi, I enjoyed that preaching to the choir a little bit, but do you have a, your background for this is Alzheimer's, but would you think that this strategy would apply more generally, or can you imagine anywhere it wouldn't apply?
Yeah, it's a very good question. Well, I think this way of thinking from a complex paradigm, in fact, is applicable to many of the chronic age-related diseases, such as arthrosis, for instance, osteoporosis, because it's, I think, having a similar root and is not, in fact, having a curative treatment yet, is also very much likely to be influenced positively by lifestyle factors and is tightly connected to aging mechanisms. So in that case, in that diseases, I think jumping to another complex system paradigm is at least helpful in giving additional perspectives.
Got a comment from Lou Lipsitz again.
Hi, Marcel. Thank you for that wonderful talk. And in fact, I couldn't agree more that Alzheimer's can be approached with a complex system approach. But I'd like to reverse the paradigm a little bit. I would argue that healthy neuronal function requires a complex system, that the complexity is in the ability of the brain to integrate a whole host of different physiologic functions. And what beta amyloid and tau might do is interrupt the normal complexity, the complex interactions of brain networks. In fact, we and others have shown that. So that what Alzheimer's is sort of the loss of this healthy complexity. And what you're trying to do with your interventions is restore that complexity by improving cardiovascular function, improving social function, integrating the various components of the brain and restoring networks so that the brain now is more complex and Alzheimer's obviously is defeated. So that's sort of a reverse of the way that you've presented it, but I think both approaches are certainly going to lead to the same positive outcome. Thank you.
Yeah, thank you very much, Lou, for this very inspiring suggestion. And I think you're completely right that you can also look at this perspective for the other way around. And probably we just have different levels of complexity that we're talking about, and that can have more or less stable states or slow decline, and also a rapid decline when there's another disease or when there's a delirium in stage. But right, you're absolutely right that you could also argue this way around.
So yeah, another thanks to all of our speakers. Again, thank you, Marcel. This is so different from the first two talks, and I think a great way to integrate just how broad the applications of complex systems theory could be for understanding of aging biology and how to use that science to help people. So let's move ourselves now to a discussion mode, and I'd like to start that. First of all, just maybe if everybody can put yourselves in the gallery mode, and just show your show your camera. If you if you're from the aging field, please turn off your camera, and if you're not from the aging field, please turn on your camera. So I just like to see how many non-aging experts we have here. So I am getting Jaap van Til, Greg Wilcox, and David Goldstick as the non-aging experts. I don't think, unless I missed someone. And so what I would like to start with is to ask the three of you to jump in. We haven't, I mean, the time for the workshops is somewhat limited, but we haven't had time to do a full introduction to aging and complex systems, but we have three great examples. And I would like the non-aging experts to start off by just sharing some thoughts, if you have any. If you don't have any now, we can come back to it, but of how your take on complex systems might inform the sorts of things you've seen today. And if you need me to unmute you, let's see, I'll have to do that, right? And yeah, let me know if you want to jump in and I'll unmute you. Jaap is trying to, sorry, this is a little hard to manage here.
Okay.
Okay, can you try again?
Do you hear me?
Now we hear you, sorry.
Okay, fine. I just wanted to have a comment from another field. It's biology. It's called Liebig's law. If you want to improve crop yield, you have to remove obstacles. Certain parameters are in the way of getting crops to grow. So what Marcel told us is, in fact, the kind of same approach, that you remove obstacles, that you reconnect things which were blocked. So it is quite a normal procedure in organizational science also now, to, instead of having very broad policies, you just go in and look where the bottlenecks are and try to repair them. So I'm very much in favor of what Marcel told us, but this has been standard practice in biology. Crop yield is because of sun, of water, of fertilizer or whatever. That there's always one thing which blocks it. It's like a congestion on the road. The first car in the congestion is holding up the rest, and if they drive faster, there's another car which steps in. Politicians never understand this, that if you haven't taken the measure, that the bottleneck just moves to another place, you know, and you have to attack the bottleneck there. Is that reasonable, what I say?
Yeah, I think it is. And I think we can get other reactions too, but my impression is this will be very promising, but a challenge in biology where people really want to understand the mechanism and intervene in it. And, and I think we'll have some convincing to do, but it may be that, at least for the time being, it's the best way forward.
Well, I'm 75, so I'm very interested to get them to succeed. And I live very near Nijmegen, where this group is based. So yes. I would applaud it when they succeed.
Thank you. Other interventions from non-aging specialists? Greg Wilcox, go ahead.
Yeah, so I really liked all of the talks today. I thought they were super informative, especially because I'm coming from like neither really a biology nor an aging background. So my background is in really artificial intelligence and in like like basically human level interactions. So yeah, what I found like really interesting was this connection between Marcel, what you presented, and sort of this forest fire model of basically emergent behavior. I don't know if anyone else has heard of this model, but it's basically like if you have a grid of trees, and yeah, a grid of trees in a forest, like a 2D grid, and if one tree sets on fire, then the neighboring tree sets on fire. And so the fire propagates from one side to another. And so I guess what I was seeing is the similarity between this model of how fires spread in a forest and the likelihood that the fire goes through the whole forest, and the ratio of trees relative to open spots in that model. And then also Marcel, what you showed, which was a model where parts of the brain get damaged and then a percentage of those get repaired. And so as you age, like in this model, more and more parts of your brain are becoming useless for specific tasks. So you could think of a signal as the fire in the forest fire model, and then the thought or the pattern of thought, the ability to complete that thought in the way that you want to, as like modeling whether that fire travels through the whole forest. So this is just a very tangential way of saying something that sparked my attention was the similarity between these two models, maybe in different situations, and that it might be an interesting, I don't know, I'm just wondering if you've heard anything like it, or if that type of idea is feels right. Sorry if that was long-winded and very strange, I guess.
No, it was great. Thank you. I was just trying to get my microphone unmuted. I don't know if also Marcel wants to react or jump in. You're still you're still muted.
I think both Jaap and Greg had great comments, and well, Greg, I completely agree that there is a lot of similarity, and I think that you could go for agent-based modeling and try to, in fact, model both the fireworks and also Alzheimer's spread over the cortex, and have, in fact, similar insights from that. Of course, the parameters will be different, but it points at, I think, behavior as an agent-based model. And also the way of blocking, what is blocking in individuals, as Jaap told, is really a logic step, but it's far from the pharma logic which tries to heal everyone with this moving the same block away, though it may not be a problem in many individuals. And well, that's the thing we try to tackle and try to shape into doable interventions that are more individualizable.
Okay.
So I'm thinking of how we can link all of this also into things that we can put in a more general summary or research agenda. And it seemed like part of the point here, I think there's actually 2 points that are coming out of this. And feel free, anybody can jump in. You might have to signal me with a message if I haven't unmuted you already at some point. But the one is the idea that because of emergence, you'll have similar presentations of things that may have very different underlying causes. And so if we want to be able to intervene, and this could be in many aspects of aging, we may need to go beyond the similar presentation of the end stage phenomenon and be looking at the variety of processes that may produce that. The other thing that I'm seeing is the idea of propagation. And so Claudio Franceschi in Aging has referred to propagaging, where one thing goes wrong and it makes another thing go wrong, it makes another thing go wrong. And I think also we can apply that perhaps more generally than Alzheimer's as well, in thinking of these kinds of cascade effects. The actual of Andrew Rutenberg's models, which do a very similar thing in terms of frailty. So if you think of emergence and cascades being the 2 messages that I'm taking. I have some people in the chat here. I'm sorry, I'm not very good at monitoring the chat and listening to people at the same time. So I'm going to read, well, everybody can read the chat, so I'm not going to read people's individual messages.
Nicholas is asking for being unmuted.
Yep. Okay. I will get you there in just a second. Oh, I'm seeing more hands here. Go ahead, Nick.
Hello. Okay. Yeah. So you said the similarity of endpoints and having to go beyond that to look under the hood, right, to see the processes. So isn't like the one conclusion from Nan Hao's work is the exact opposite, which is when you made the intervention, you got a third new path that was actually not predicted by either of the two paths present in the wild type. So I mean that's an example where looking under the hood actually does not predict the outcome of interventions.
That's certainly true. So I mean, I guess the question then would be, I don't think we need to be in a mode of one of these solutions is for all situations, right? I think they can both be true. And
it's a real puzzle about what the relationship is between change that affects the outcome and change that affects the wiring of the system and the processes that are emerging from it.
Right.
Like that's like, and I don't know a way of predicting whether the two would be linked or not in a living system. You know, I could imagine some cases where there's some canalization at the process level so there's always a link between the processes and the outcome. But I could also imagine a situation where it's just very chaotic. There's a whole bunch of nonlinear effects, and it's, you know, knowing the system is, it's just like predicting the weather. If you change a pressure system here, it's really hard to figure out what's going to happen globally at the end of the week.
Right.
And I don't know if we know enough about aging to know how predictable the outcome should be given the processes.
Right, and probably in some cases, some parts of it may be predictable, and some parts may not, right?
Yeah.
I think, I mean, just thinking off the top of my head, it would seem like those two things will, it will actually be very informative to see what we can predict and what we can't, because what we can't predict is probably representing some like noisier aspects, whereas what's more predictable is canalized and more biologically functional, perhaps.
And can the complex systems perspectives contribute to trying to bring some structure to understanding, you know, whether something's very chaotic and unpredictable, or whether something you'd expect there to be, you know, canalization to find.
Right.
So this might also give us some modesty towards expecting immediately good effects of single agent therapies. I think that's also a message from complexity science to the aging and the geriatric perspective, because often we still focus on just single agent interventions.
Yes. I think this is actually a critical point as we develop the position paper to make, because I think it will very strongly make the argument that if single agent therapies aren't working, the way to get to things that work is to do this. Andrew had a comment as well.
Yeah, just reflecting on the three talks, and we had talks about epigenetics and single cell level, and very much at the Alzheimer's level. And I think that there's this strong sense of multi-scale that people are agreeing on, and I think that would be true in any issue, Alzheimer's or any other aspect of aging. And so if we're thinking of complex systems modeling, it needs to be something that you can apply to, say, you know, methylation data and single yeast data and Alzheimer's data. And ideally, you'd get data from all three different regimes that you could incorporate, but not necessarily. So I think that if we're going down the paths of making separate types of models for these different phenomena, then it's going to be hard to plug them together as new data emerges. And I think that if we can find modeling approaches or complex systems modeling approaches that can incorporate different types of data, then I think we're going to get new insight. And so a related point to that is if you get Alzheimer's, you could still have mobility issues. You could still have, diabetes. You can still have, and so if you have a model, someone has a model for diabetes, how do you fit them in your Alzheimer's model? It's challenging, and it's scary. What Lou said, just to finish, is if we also have to have a model of healthy function, then we're sort of doing the job of all of biology, and that's a little intimidating. Yeah.
Well, as intimidating as this can be, that's really key, understanding the complexity of health. I think the mathematicians sometimes talk about the percolation mechanism, where you have a complex system where you have many, many elements, they interact with each other to maintain homeostasis, and then you start picking them away one by one. And the collapse of the system is nonlinear. At some point, it starts accelerating because you have reached a critical threshold. And if you imagine a random process where you do not select these elements, but you take them at random, the curve of the sky will be different. It's a little bit like, the two, elements that Nan was describing. And instead of thinking about two, you need to think about thousands. And I think that the key is certain, these elements are more critical than others. That is where we need to find out what are the nodes that without which we will have faster collapse. Because otherwise we can't really handle the complexity of thousands of interaction. And I agree that therapeutic intervention will require multiple, type of drugs or intervention. There's not going to be 1,000. They're going to be 3, 4, 5, 6, 10. We can imagine 15, but we can't really replace, or recreate, 100 of functions. So understanding what are the critical nodes that we need to intervene on still remain very, very critical. We can approach them and understand them, from the top, from the complexity, and simulating a reduction of complexity, understand which one of these nodes is more important. I mean, for mitochondrial function, for example, that mitochondria are a network in the muscle. But if you do a mathematical simulation, you will find that if you pick certain nodes, nothing really happens because around it, there is a way that the network compensates. But there are some critical nodes at the, close to the Z line. Those nodes, if you pick them enough, the network collapses immediately and it's no longer able to communicate. And so I think that that's where, to me, the complexity can gain something.
This is Lou. I have a comment on both of the two previous comments. Regarding diabetes and other systems, each needing its own complex system to explain it, I think we have to move away from these disease notions. Diabetes is impaired glucose metabolism, and it is not just in the diagnosis of diabetes, but it occurs in other systems and processes. We've learned recently that the ACE gene, the angiotensin converting enzyme, is not just all about kidney disease and hypertension, it's also about COVID. I mean, these are not, we can't be so reductionistic that we think of one disease, one system, one interaction. We have to think more functionally. So as a geriatrician, I think of outcomes not as disease so much, but as function. Why can't somebody walk up a flight of stairs? Well, I mean, that has something to do with mitochondrial energetics. It has a lot to do with muscle, with brain, with neural integration. So I think we have to think systematically across multiple systems to really get a grip on this issue of complexity and how it impacts aging. And with regard to Luigi's comment about finding a critical node, I think of a complex system in our bodies like a highway. If I want to go from Boston to New York and the turnpike is blocked, I can still get there. because it's a network and I can take a different pathway. And I think it's the same with the body, that we are compensating constantly and adapting by taking different networks and different approaches. And it may not be so simple as a critical node that we're looking for.
So
Thanks.
what I'd like to do now is to share my screen, where I'm going to, if I can find the right document here, so this is some of what we want to discuss tomorrow for the paper, but I'd like to start setting that up. We have about 10 or 15 minutes left, and I'd like to structure that in terms of trying to figure out what are the key subjects or elements, because if we want to have a program or a research agenda, we have to be at least somewhat limited in what we talk about. And so I've taken careful notes of what we've said up to this point, and most or all of that will make it in here one way or another. Let me go very, very briefly over what I have, and we can start then to think through what's missing here, essentially. So for me, a starting point is to think about what is a complex biological system, because obviously aging needs to refer back to that, and what's unique about biological systems. So natural selection, homeostasis, multiple parallel hierarchies. We often have sparse data challenges in biological systems. And then how can this perspective change the research questions we ask and make for better aging? So we had, kind of tentatively come up with network structure changes, cell fates, search for emergent phenomena, bow tie structures, which I don't want to get into now if it's not familiar to people, and then methods, so critical transitions and how we can predict those, mapping and applying causal networks, inferring state dynamics with sparse data. We've already listed things like emergence, cascades, multiple pathways to, as Lou was just saying, there may be some critical nodes, but there may also be ways around those critical nodes. So what's missing here? And let me make sure I can see if people are signaling me. Okay, so Martin has a comment here. Go ahead.
Yes, hi, thank you. Yeah, this is a great discussion and so important. I think the talk by Marcel and also the presentation by Han on yeast cells makes me wonder if we agree that there's individualized trajectories. So there are multiple ways for cells to age, and there are multiple ways for people to get Alzheimer's disease. It makes me wonder how much of this we want to take into account. And because I think an implicit assumption is that there's one way that people age, and then we're trying to find that one way, either looking for critical nodes or other things. And there's also a limit, on the other hand, if everyone ages in a completely different way, then it's hard to do science around this. So I think this idea of individualized trajectories or individualized set of causes, of equifinality, multiple different types of interactions can lead to the same outcome, is something we need to think carefully about, and how far we need to push this. Is there 5 ways to get Alzheimer's disease, or there a dozen? Is there 100? And same thing for aging. Is how many different trajectories or strategies are there to either age or not age?
Yes, thank you. Yeah, I certainly agree that this is a crucial question. I put in now heterogeneous aging and equality. Jaap has mentioned that we should have more on network dynamics. I'd like to throw out a question here too. I mean, I think nobody would doubt that networks are crucial in understanding biology in general and aging specifically. I think though that there are challenges, and one of them is that we can't map the whole network yet. We're actually quite a long way from that. And to some extent, on smaller scales, some network approaches have been very, very fruitful. But to what extent does the challenge of how far we are from mapping biological networks mean that this is a promising solution or not?
If I could, this is Andrew. You know, Nan Hao, in his first talk, he had a network. You know, he had a two-component network. He didn't phrase it that way. He phrased it as, you know, coupled differential equations. So I think a network is an organizing principle. It's not necessarily the end of the story. And he showed that that was quite helpful in a yeast system, even though it was missing a great many of the genetic components of yeast. So he's already shown it's useful, even in a very reduced circumstance. And I would tend to think that that conclusion generalizes to other organisms. But the key thing is that, you know, just drawing a network with some arrows isn't anywhere near a solution.
Right. So maybe what we're coming to is network dynamics with targeted approaches, right? So the goal here is not necessarily to map the entire network of everything. It's to judiciously use network approaches where they can be applied, given, you know, either because we know a subnetwork very well, or because we have, I mean, like your work is a great example as well. We don't have to know the network necessarily to be able to infer some of the properties of how networks would work at a frailty level.
Yeah, and we're trying to move past that. But I guess my point would be that fleshing out the network, a Barabasi style network of links and nodes, you have to embed that within a broader, you know, dynamical picture for it to be maximally useful.
And I want to add a little bit to that point about the network dynamic, and I totally agree that given that what we know, it is a rather very challenging approaches. But I think one important point, I think Nick also make it in the comments in the chat, that the reason that we can have our network working, we can understand how this network works, even a small network, is the capability to mirror the nodes, the important nodes in the network over time. Because how nonlinear dynamics can help us is really to describe and simulate and predict how this system, although it is very limited, how this system evolves. And because of the parameter and all these things, it may go either many ways. Being able to experimentally track each individual nodes in this small network and really see how it works, it's quite important for really using the nonlinear dynamic model to understand the system. That's actually why we start with yeast, because when the complexity of the organisms start to go up, the complexity and the kind of model we can do and the kind of measurement especially we can do become very limited. So I think this just add some comments to that.
Yes.
I have a comment to make.
Yep, go. Sure, go for it.
So I'm just wondering, you know, if there's a good idea for why we should study aging, you know, within the framework of complex system. I think that's an important question, you know, to think about the goal. And the other thing is that if we do this, what is the complex system we're looking at in the context of aging? And I don't think, you know, I've heard an answer to that question today.
Right. So I think my talk tomorrow will address it a bit at least, but I think there are a couple ways we can look at that. So one is aging is a large field, complex systems could be applied in a lot of ways, and there might not be a single answer to those questions. But another way to look at it is that, and this gets back to the question of do we have to look at everything that is health and take on the job of all biologists? And you know, I think increasingly I see people in the aging field increasingly moving towards what is health, but you know, in some sense that might actually be the question, because if we're looking at the converse of health somehow, then we might need to tackle all of it, and we might be the part of biology that is kind of at the forefront of asking that question in a broad sense.
I think it's a good idea. I mean, and I really like, you know, what Louis said earlier. I mean, he did talk about health, and you could think of health as, you know, something very complex, but it has, in the context of aging, you know, there are certain trajectories that perhaps you will not find with other, you know, problems with health. So
right.
I think these are important questions. You know, these are about definitions.
Right.
And if you don't have a proper definition or a clear definition for what you're looking at, then it's very hard to think about how useful this is going to be.
Right. So make out here to circumscribe our task.
What also,
and another thing that I wanted to mention quickly, perhaps, is that, I mean, what we saw today is the complexity in terms of the different scales and the different types of questions that are asked at those different scales. And if you want to provide some answers that are sort of generally applicable, then you really need to answer the question of how you establish bridges between those different scales. And that is a problem of complex systems.
Yes. Yeah,
and therefore we might also use, in fact, generic models, such as there are animal models for therapeutics. You might also need simulation models for complex systems, so that we, for instance, can test resilience of health for different stressors and have models available that can be applied for different disciplines. In fact, that's still lacking, I think.
Also making some notes here from the discussion. I
think the other thing that was kind of brought up a little bit by a few people that I don't know if you wrote down here is the need for longitudinal data again. Like, I think Nan brought it up, but it's, if we actually want to study, I think that's the hard thing with Alzheimer's disease, right? Is you can't, most of it, we're studying it after, you know, post-mortem, looking at brain tissue. But to understand the process of how the system's changing, you're going to have to somehow figure out how to study some of these things through time, which I think presents a challenge in terms of modeling.
I've got a note here that I think we have to close because in 3 minutes the next session is going to start. So we're going to stop there for today. We'll pick up tomorrow. To my co-organizers, I didn't get firm responses, but let's try to meet for noon tomorrow. I'll US Eastern Time. I'll send you an invite for that. And I'll see you all tomorrow at 2:30.